Welcome to the inverted laser scanning microscope. It's called Zeiss LSM800. It's located in room 355. And please take care of this lab is also used for other purposes, so that's why we have curtains, and you can handle the light separately, so you have complete darkness in here with the curtains. So the startup procedure is written on the wall, and I'll go through it in this short video. First of all, you remove the cover, and then you power on the system. First power button here, and then we have some power buttons over here. Okay, so now you see all the hardware components of the system, and in the top you see a stage control power supply and the wide field fluorescent lamp. And there's a reason for the tape. It's because you never need to touch these ones, so don't shut them off. So everything is controlled down here. So after starting up the power on the main panel, you turn on the system and you turn on the components, and then you can see they are all starting up. This one is the laser control, and when you turn the key, you see that the laser is on. So they are diode lasers, and they are not generating a lot of heat, so they don't need to heat up and cool down as you may be used to. The last component is called the area scan, which is a special component or a special detector for better resolution than normal LSM. Okay, and then you turn on the PC, and while you wait, take the opportunity to write down in the notebook that you are here, and also if there are any issues, please write them down for me and the next users to know. There's also a short guide located next to the screen. You start the system as LSM user. Then you map your network drives, as you do on all the other systems. And you turn on SendBlue software. So now you have the choice of using this as image processing, but you can do that elsewhere, so please use this as an acquisition system so you're not blocking for other users. So after hardware initialization, the system is ready and starts out in the locate tab. So now we are in load position, and you can always go back to work position, and you can also set a new work position. So now I'm in the load position where I can safely handle the microscope. I take back the illumination pillar, and then in this case I take the whole insert out just to show you, and you can, of course, you keep it clean always, no oil on this system. You put in your sample upside down because it is an inverted system, so you can have it like this, and you see the red dot here needs to go to the red dot on the XY stage, and you put it something like this, and then you slide it in. So we have here the touchpad, and you can use it to locate your cells and also to make easy touch changes on the microscope. So we already went through the load position, and always remember it so you're not damaging the objective lenses. Then you have the illumination. So this is a transmitted light illumination on and off, and the reflected light illumination used for wide field fluorescent illumination, just to locate your cells. It's already on. You have also this make it visible. So touching that, then you can see the settings is sent to the oculus, and you can see your image. Going to the microscope control, you see the objective lenses here, and the filters, and the light path, and so on. Here we have an overview of the filters: 5, 10, 40 dry lenses, 63 oil lens. Then you can go to the reflector, and you have DAPI, and you can hear it's changing filters, and GFP, and the red, and then the far red. which you cannot see by eye, so you will focus on these three. The same functionalities are on the microscope. Yeah, so first of all, as in most microscopes, you have fine focus here, coarse focus here. Then you have transmitted light shutter. You can see it changes also over here. And reflected light shutter, same function as over here. Then touching this one is going to the load position and touching again, or this button, then it goes back to work position. There are also buttons down here. This one goes to the change objective, so one side and the other side. So these are just fast tools when you are sitting by the microscope. Also on the left side you see filter change, and you also have some other functionalities with a condenser. Okay, so for transmitted light you will want to image using differential interference contrast, so also called DIC. So DIC, you need to pay attention to align it correctly. And don't put too much pressure on it, so hold your hand on the XY stage. I'll show you, some people put it like this and they have a very bad experience of imaging because it will be not firm. So slide it in always. You don't need to take it out because you can just take out the sample, but you may have to take it out at some point. So I take down the illumination pillar again, and then I move my joystick to the sample position. Then I go back to the work position and I go and control the microscope. So I have the different objective lenses, so 5, 10, 40 are dry lenses on this system, and then there's one oil lens, the 63x oil. First of all, we need to turn on the reflected light illumination. So you can turn it on here, or you can also turn it on in the software. And if you are using transmitted light, so light from above through the sample, then you also need to control that. Okay, the XY stage control is just like a joystick, up and down and so forth. And it has two modes, so now it's in the coarse movements, and pressing this one it'll do fine movements. And back to coarse again. After the software has initiated, you start and see the software like this. You can resize it so it fits to your vision up here in the right upper corner. Then you have these tabs called Locate, Acquisition, Processing, Analysis. In this video I will show you Locate, just to find your sample, and then the features you have in Acquisition to actually acquire your images. So you simply go from top to bottom. You first start out turning on the lights, so the transmitted light source is on already. The reflected light source for wide field fluorescence, so if you want to check your DAPI, FITSI or whatever channel just by the oculars, you need to turn on this lamp. You can see it down here. This is the lamp down here. So here we have the diagram showing the light path. So we have the sample here, and if you want to locate it using the bright field transmitted lamp, you turn it on, or you increase the illumination power here. I want to start imaging with 10x just to localize my cells. I could also use 5, 10, 40, and then go to 63x oil lens if I want the best resolution and quality. Pay attention to all these are air objective, 5, 10 and 40. 63 is with oil immersion. So I showed you already the lamp. The lamp is on. Here we see the shutter is also open. We send in 100% of the light through this attenuator. We can change that if we want to. Here we also have a reflected light shutter. You can turn it on and off. You can also do that on the side of the microscope or in the touchpad. And here we have a field stop, so you can decrease the field stop to change the contrast. Normally just keep it open. Right now I do not see fluorescent light, and that is because there's no filter cube in it. So we can start out selecting one of these. So in my case I have DAPI and I have Sci5. So before you're able to visualize it, you need to put all the light to the oculus. So you simply need to make sure that all the lines are going from the light source through the filter to the objective to the specimen and back down again to the ocular. We are only using this locate to find our focus and find our sample, and then we are going to this tab with the acquisition mode. And at this point we use the lasers and we use all the confocal parts in the box on the left side of the microscope. This tab you can choose an experiment. You can, if you have made something already, it's displayed here. You can also open a file which you acquired last time at the training or whenever you did something, and press this one to reuse the settings. So in that case you will actually be able to compare the data. Next button is called Smart Setup, and by hitting that you get some help setting it up. In the Smart Setup you have two detection modes. So you can either go for LSM Confocal, which is the normal way of doing confocal imaging. You can also start out using area scan, also called SR for super resolution. So you can already choose area scan imaging at this point. I'll start out doing normal confocal and I'll add some channels. So I add my Alexa 488, 647 and DAPI. This is a standard way of doing some imaging, 3-color, and you can of course add more. I'll just show you this. And in case you need more, I'll open it again. These are the recently used. Höcht. If you want to take that instead of DAPI. DT Tomato or Sci5. So I of course don't want all this, so I'll delete some of them. So in this setup I'll show you a green, blue and a far red channel. And it's also showing quite well that you can do this numerous ways, and not all is optimal. So let's go for the first one is showing the fastest. So you see the speed bar here is 100%, and it's the case because everything is acquired in channel 1, so it's only acquiring with one detector, the red, green and blue. And it's not putting the cutoffs very precisely, so it's you see some blue in this negative bar in the green area, meaning that you have some bleed through from the blue and tiny bit from the red into the green. and also in the blue bar you have something from the green. So this is not what you want to do. Next one is best signal. So best signal you use independent channels for each color. So you have 1, 2, 3 here with a red, green and blue. You see the gates are defined here, and you have very minimal crosstalk. So this is good quality, and unfortunately not as fast, of course, because you are using 3 detectors. Something the software calls the smartest is detecting with 2 channels, so green separate and blue and red into 1 channel. So you see blue and red will not crosstalk, so you can easily acquire them in the same channel. This will give you relatively high signal, so you have around 70%, and in the best signal you have around 90. So of course it may matter to, if you have weak samples, you want to go for best signal, but most samples or many samples will be sufficient to acquire with 70%. Because what you gain here is speed, so you have 2/3 of the maximum speed using 2 detectors rather than 3 detectors. In this test sample I don't even have Alexa 48. I'll delete it, so I will have only far red and blue. I'll show you the best signal in this video. And you see here what we just did in the smart setup was to determine an imaging setup. This can also be done manually. So here you can see you can go to area scan, or you can go to lambda scan, but we say in the normal Confocal. You can switch the track every line or every frame. You can do it in the fast mode also. You can put the gates here, the cutoffs. So in the red channel, you can change it if you're not happy with it. So simply do like this, and then you go to track 2, and you can do the same here. So in case you had more channels, you could manually go in here and decrease the detection window. And so all this I will go one by one down here, and when we actually imaging, then I'll use this part. I'll come back to that. So here you can see the area I use. So I'll just simply use 1x area, but you can crop. So in case you're only looking at minor parts of your detection area, then you can crop this part. And of course it gets faster. You can also change to line scan if you want to only scan lines, and go back to 1 if you want to go back. I start out normally using 512 by 512 until I have my settings correctly. At this point I'll go higher, pressing optimal. So while optimizing, you don't want the best quality because that will take time. You can easily go with a 512 by 512. So I'll already at this point go to stick to 3x oil. So you stick to 512 by 512 to start out with. When we are actually imaging, we can press optimal, and then it will select the optimal pixel frame size according to the objective in use. You can press this presets to go back and forth. So I go back to 512 and then I optimize. Next thing to change is the scan speed. So if you are keeping this very high level, maximum is 9, and it gives you a pixel time or pixel dwell time at around 1 microsecond, which is rather short. So you will increase the quality in the image by decreasing the speed. So keep it at 6 or 7 will give you much better image quality. So the dwell time around 2 microseconds, or even 4 or 5 would give you super quality. I'll keep it at level 7 at this point. Then the direction of scanning, so you can tell it to scan the normal way, so scanning from left to the right and jump back left to right again. Or you can do a two-way scanning or bidirectional, so it will scan from left to right, right to left, and so it will double the speed. So normally I use it. It can give some artifacts, and just pay attention to the image quality, and don't forget to correct. So you can auto-correct, leaving the system for some seconds to scan. So it's rotating the scanner head and aligning it. So you don't need to do anything more than just pressing this auto, and it will determine the best way to do it. You can see here the process, it takes 10 seconds. Next step, you can interpolate, so you can skip some lines. So you will do that to increase the speed, of course. I'll keep it at normal without interpolation. Then averaging is something you do to limit noise. So you can acquire, in this case, 2 images and average them. So if you have noise, little noise, you will cancel it out. Especially when you go to 4, you will have a good effect of averaging. And going from none to 4 is a really good tool to get rid of noise. Of course, you can go up to 8 and 16, but that will require 8 times the time or 16 times, so you'll waste a lot of time. I'll go back to none. When you do this, you can do it repeated by line or by frame. And normally you use mean intensity. You can, of course, have reasons to go to some intensity if you need to sum up the intensity of the image. Next thing is the bit depth. So you can choose either for 8 bits or 16 bits. So that's the number of grayscales you will have in your image. So 8-bit means 2 to the power of 8, which is what we can see by eye. So if you use the images just by to represent images, you don't gain anything to increase bit depth. But if you want to quantify, you should go for higher bit. So that this is 2 to the power of 16 grayscales, which will be useful when you are analyzing it with a quantification software. Here we are at our 2 tracks. So we have track 1 showing Sci5, track 2 with a Höcht. You can deactivate the tracks here, activate again, of course, and then keep them. The gray is highlighted as the one you will show on the screen in a minute. So now track 1 is activated, and you see you will be using laser for 640, and the laser power is always starting very low. You can increase it by this ruler or simply by adding number. So usually 1 to 2% is enough. In case you need high power, you can activate this one. You will have a warning because you often don't need to go to high intensity range. But in this case, I can then go on all the way to 100, and I'll probably burn my sample if I do it. So I take it off, and then I can only go to 5%. And I think 1% is enough. Next thing is the pinhole. So the pinhole is determining the confocality. So reducing the pinhole to a certain size given by the objective length and the wavelength. You can go to 1 area unit. So this diameter of the pinhole gives you confocality. Of course, you can still have some blur, or you could have very little intensity. So that'll be the reason for either going lower or higher. If you go all the way open, or you can use this one, then you don't have a confocal system. So then there is a wide field system, and you will see all the intensities from the whole section. Then we have the master gain. So that's the intensity of the signal, so the voltage amplification. That is normally in the range of 500 to 750-ish, 800 maybe if you have a weak sample. If you go higher, then you also increase the noise dramatically, the background. So keep it at maybe 600. Then when we see the image, I will show you what digital offset and digital gain is. Start out here pressing live. Then you can also hear the scanner is going in a fast mode, so it is scanning the fastest possible way. And I see my actin here, so I can scroll by the normal, I can focus by the normal focus wheel, or I can press Control and scroll with the mouse up and down here. I'll deselect it again and tell you the difference from live to continuous is that you go from the fastest acquisition setting to the settings that we actually set here. So all what we did up here with the speed and frame size and all that is applied. And we see it looks fine, but we do have the whole histogram displayed here. So it's meaning that we have some saturation. You can tick this one on, the range indicator. Then you see the blue is darkness and red is oversaturated. So you don't want oversaturation, so we should go down either by the different means we have. So the laser power or the master gain would be the things to change here. And the general notion is that use more laser power and less gain, you get the best image quality. Of course, you may have reasons to go low because you may have a sensitive sample, so phototoxicity is of course increased if you increase the power. But here I only use 1%, so I can easily go down to, say, 500. And then we see the histogram here going from black to white. We only have very few peaks out here, and we don't see any red dots anymore. We can take this on off, and we see a quite dim signal compared to before. We can change the display by hitting minimum, maximum, or best fit. Then we can see more of our signal, but we don't have, of course, saturation because we don't change anything by changing the display. We simply change the way we see it on the image. Okay. So now I'll go to my second channel. So that's the Höchst channel, and I'll deselect Sci 5. Right now you probably wonder why don't I see anything on the Höcht. First of all, I show all the display, but I only have something in this region. So I can hit Minimax, then I do see some signal. It's weak, and it is weak because I'm using 0.2%. Let's go to 1% like we did before. And then we are using more of our histogram. Still, we could use more, so let's go for 2%. Then we see we use almost all of our histogram, and the image looks very good. Okay. And also in the track 2, I will press one area unit. Then we have our digital gain and digital offset. I'll show you what that is. It's not needed in this case, but digital offset is where the software is recognizing zero or darkness. So you can increase it, then you get everything. So you see what was before darkness was you can actually see tiny bit of signal. This is extreme, but the useful range would be just around 0, so a few up and down. In this case, 0 is perfectly fine. Digital gain is you can enhance the signal. You can see what is happening. You get very high signal, and you also increase the background. So mostly you don't need gain. Of course, you can have a weak signal, and gain can be useful. Let's go to the other track here. So you can activate both tracks and go continuous. And we see both of them. So the other track here. Here we can play around a bit with the offset. So here you can see if we go higher, then we get rid of all what was darkness before. So this may be useful to go for digital offset of 1 in this case. So we have only very few spots that are completely dark. However, 0 is doing the job just fine. Alright, now we're ready to image, so don't forget to activate both tracks, then we can take a snapshot. So this was one image, and it is in 512 and 512 still. So that's the last thing I normally do, is to go optimal or to go to any presets that you want to use. And you can see the speed, of course, goes down when you use the optimal frame size. Alright, for the next steps I will show you just for doing it more rapidly. I'll go a bit down in frame size, then I'll do some set stacks. And also for that, I don't need the whole area, so I will just reduce the area a bit. Oh, like this. I can navigate on the screen doing like this, or I can double click somewhere I find interesting. And I can also change the scanner, so that may be useful if you have some, say, tissue or something that it matters how it's oriented. You can go back to 0. So let's just do it like this. And then we go down to our... So whenever you activate something down here, up here, you can see it popping up in the blue bars down here. So I go live to see my structural protein here, and I press Control and scroll up. You can actually change the speed here if you want to go faster or slower. So now I'm at the top, and I'll define that as my last position. Then I'll scroll down, and I define that as my first position. Just to show you, over here you can see the range, up and down. And then we press optimal. So that would be 16 slices according to Nyquist. Of course you can change it if you don't want the optimal. If you don't have time for that, then you simply do it the way you want to do it. But whenever you have activated something here, then it's no longer a snapshot, then it's experiment. So press Start Experiment. Then you see it's going slowly through the stack, and you can see the process here. It will take, yeah, it's a minute or something. Yeah, 1 minute, 8 seconds, you see it up here. So now it's done, and we have our C-stack displayed over here. We see it right now in 2D mode. We can see it in split mode, so red, blue, and then the overlay. Also in gallery you see a number of images. Maybe this is more useful. You see the stack in the top and to the right. Then you can go through the 2D representation, and then you see the 3D representation up here and down here. Also you can see a cut mode and intensity mode and a 3D mode. If you do thick stacks, then a 3D mode is of course better. This is relatively thin section, but you can see what it's able to do. You can press these functions and go around, fly through, whatever. They have some different tools. You can see them, histograms, colocalization, and you can go through them. But we stick to our acquisition, so we, instead of C-stacks, I want to show you one last thing with C-stacks. You can of course also, instead of first and last, you can change from center, so you can define the thickness of your stack instead of defining top and bottom. Most often it's convenient to use this first and last. Then you can also press this one to optimize sectioning and step. So that's something you want to do if you want to do colocalization or quantification analysis. You want to make sure that you have optimized your pinholes in the two different colors or channels. So by pressing match pinhole, it will match it according to the wavelength in the two channels. Okay. Instead of C-stacks, I'll do some tile imaging now. Then you see this tab here. You can do some simple 2x2 or 4x4, and then press it in here, and I'll go live just to check that I'm in focus. and I'll start an experiment. So it will require 4x4. Okay, so now it's done doing 4x4 tile scans, and we see the sectioning or the alignment of the tiles are quite okay. And this was a simple part of the tile view. You can delete this one, and we can go in Advanced tab. Then you have the option to go to start a preview scan. So let's go to 10x. I start a preview scan here. I went to 10x, increased the laser power, also the pinhole, because what I want to do right now is just to have an idea of where my samples are. Then go also high gain, so we can see some of our cells. So we can do a 2x2. So I delete this one. So this 2x2. And it will start a preview scan. Then we go to our 63x again, and we don't forget to go down to this was 2%. We go continuous, then we see how it actually would look like. So in this preview tab you can see a large area, and that could be tissue, that could be anything, or it could be cells where you're looking for some rare expression, and you can then define a region, let's say 2x2 tile, and you can add it here. If you could see something in another position, or you want to increase the number of images, then you see 42 images here, or decrease it. And you can also put another one. So I'll ask it to acquire these two positions. And you see the positions popping up here, or you can see them out here. So this was tiles. You can do time series. Then it pops up with a duration of number of cycles, and interval you want to wait a number of seconds. Or you can also do different bleaching experiments, and one thing that is relevant for all users is this autosave. So clicking that, then you see the autosave folder here, so you can save it locally, put it on the D drive, data drive, make your own user profile, and after acquiring, you move it to your U drive. When you want to use AriScan, it's out of the scope of this video. I'll put it on a video soon. And also you can do lots more, but this was the basic principles. In this part you go and you can do some processing. You can do this either on this computer or another computer with the Zen software. And you can do some analysis. You can quantify and do some segmentation and stuff like this. Okay, I'm done imaging, so I will go out and put it in load position and clean up after myself. We're done imaging and we want to leave the system clean and shut it down, ready for the next user. So first thing, go to load position and the objective lenses go down so we can safely remove the sample and clean it from oil and put in some ethanol on the lens paper and clean it in circular movements as you're used to. And you can go back to work position, put down the illumination pillar and close down the software. And the PC. Then you need to shut down the laser by the key and also the components and the system, and finally take the main power button